Let's begin with a simple but powerful idea. Every medicine we take is expected to be safe, effective, and reliable. But during its development, scientists often discover tiny, unwanted chemicals hidden inside the drug. These are called impurities. Sometimes they are harmless, but sometimes they can affect the drug's safety or performance. Impurity characterization is the scientific process of finding these unknown substances, understanding them, and ensuring they don't pose a risk. In this presentation, we'll explore what impurity characterization is, why it matters, and how pharmaceutical scientists solve these molecular mysteries. First, let's understand what impurities actually are. An impurity is any unwanted chemical entity present in a drug substance or drug product. These impurities can come from several sources. They may originate from starting materials used in synthesis. They may form during side reactions. They can also develop over time as the drug degrades. Or they may remain as residual solvents after manufacturing. Even tiny amounts can have a significant impact. Think of it like noise in a music recording. Even a small amount of noise can affect the clarity. Similarly, even trace impurities can influence drug safety and effectiveness. Now, detecting impurities is not enough. We must understand them, because the first priority in pharmaceuticals is always patient safety. Some impurities may be toxic or harmful. Regulatory agencies like ICH, FDA, and EMA require impurity characterization above certain limits, typically around 0.1%. Characterization also ensures drug quality and consistency. Without this data, regulatory approval is not possible. So impurity characterization plays a crucial role in bringing safe medicines to patients. To understand impurity analysis clearly, we need to distinguish three important levels. Detection is the first step. This means observing something unknown, like seeing a peak in an HPLC chromatogram. Identification is the next step. This means determining the exact chemical structure of that impurity. Characterization goes even deeper. It means understanding the impurity's origin, properties, and behavior. In simple terms, detection is seeing a person. Identification is knowing their name. Characterization is knowing their full story. This complete understanding is essential in pharmaceutical development. Scientists use several advanced analytical techniques to characterize impurities. HPLC is used for separation and quantification. It tells us how much impurity is present. LC-MS provides molecular weight and molecular formula information. This gives important clues about the impurity's identity. NMR is considered the gold standard. It provides detailed, three-dimensional structural information. FTIR helps identify functional groups present in the molecule. Together, these techniques allow scientists to fully understand the impurity. Let's look at a real-world example. During a stability study, scientists noticed a new impurity at 0.3% after 3 months. This raised concern. They analyzed it using LC-MS. They found a mass increase of 16 units. This suggested oxidation. To confirm, they used NMR. NMR showed oxidation at an aromatic ring. Now they understood the impurity. To solve the problem, antioxidants were added to the formulation. This prevented further oxidation. This is how systematic analysis solves impurity mysteries. Once an impurity is characterized, the next step is qualification. This means evaluating its safety. Scientists perform literature reviews and toxicology studies. They also use computational tools like QSAR modeling to predict toxicity and mutagenicity. Based on these results, Safe limits are defined using ICH guidelines. This ensures patient safety while maintaining drug quality. Impurity characterization comes with several challenges. Some impurities exist at extremely low concentrations, below 0.01%. Detecting them requires highly sensitive instruments. Isolation can also be difficult. Impurities may degrade or overlap with other compounds. Some impurities have very complex structures. Understanding them requires advanced NMR and expert interpretation. This makes impurity characterization both challenging and fascinating. Impurity characterization plays a central role in pharmaceutical development. It helps optimize synthesis processes. It ensures drug stability. It ensures regulatory compliance. Impurities above the identification threshold, typically 0.1%, must be fully characterized. Without this information, regulatory approval cannot be achieved. Impurity characterization provides the scientific foundation for safe and effective medicines. To conclude, impurity characterization is the science of understanding the unknown. It ensures patient safety. It ensures drug quality. And it ensures regulatory compliance. Every impurity tells a story. And through scientific analysis, that story is understood. Because in pharmaceutical science, patient safety begins at the molecular level.